Welcome back, grade 9 students of St. Paul American School Clark, to today's Explainer, G9 Biology, Unit 3, Lesson 6, Gene Regulation and Mutation and Genetics and Biotechnology. I know, that is quite the mouthful of a title, but get ready because we're going on a high-energy journey right into the very code that makes you, well, you. Think of your DNA as the ultimate biological instruction manual. Today, we're going to learn how gene regulation acts kind of like a reader, choosing exactly which chapters to read. We'll see how mutations act like sudden typos in that text, and then how biotechnology gives us the literal power to pick up an editing pen and rewrite the code ourselves. Let's dive in.
Okay, let's quickly check out today's agenda. We've got 1. Lesson objectives. 2. Regulating the genes. 3. Stickleback fish analysis. 4. Modifying with biotechnology. 5. Mutation misconception check. And finally, 6. Formative check and recap.
Section 1. Lesson objectives. Let's start by establishing exactly what we want to master today.
So by the time we wrap up, you should walk away with a solid grasp on 4 main things. First up, how genes are essentially turned on and off. Second, how a very specific mutation in a gene called PITS1 actually affects fish out in the wild. Third, we want you to understand exactly how groundbreaking biotech tools like PCR and CRISPR actually work. And lastly, we're going to correct some super common misunderstandings about genetic mutations.
Section 2. Regulating the genes. Let's see how your body decides which genes to actually use.
So, here's a totally fascinating fact. Virtually all the cells in your body have the exact same genetic makeup. So why does a skin cell look and act completely differently from a muscle cell? Well, it all comes down to gene regulation. Imagine a huge house, and the same electricity is running all through the walls. Regulatory switches are just like the light switches in the different rooms. There are specific regions of DNA that control whether a gene is turned on, meaning it's transcribed, or turned off, in different tissues at different times.
Section 3. Stickleback fish analysis. Let's see these genetic switches in action using a really cool real-world example.
What's wild is that a regulatory switch doesn't even have to sit right next to its gene. It can actually be pretty far away. The DNA strand literally loops around, which allows these special proteins, called activators, to bind right to the switch. Then mediator proteins basically bridge the gap. They help trigger the RNA polymerase at the promoter region so it can actually start reading the protein-coding part of the gene. It's like reaching all the way across a room to flip a switch that turns on a lamp on your desk.
Now let's apply this to the stickleback fish and its PX1 gene. This specific gene is absolutely crucial for building body parts, and that includes the fish's pelvic spines. Now, in some freshwater stickleback populations, there's a natural mutation that actually deletes the specific regulatory switch just for the pelvis. And because that switch is totally gone, the fish doesn't grow pelvic spines. But here's the amazing part. The fish survives perfectly fine. Why? Because the main coding region of the PX1 gene is completely intact, and it still has working switches for its jaw and its pituitary gland. It's a targeted change that affects just one single part of the body.
Section 4. Modifying with biotechnology. Having seen how nature masterfully regulates genes, let's explore how scientists step in with their own editing pens.
Let's start with a really clear definition. Biotechnology is this incredible field where we use technology to intentionally change the genetic makeup of living things for human purposes. This is huge. We're talking about anything from creating crops that naturally resist pests, all the way to developing revolutionary treatments for genetic diseases. We are going to look at 3 absolutely key methods scientists use to do this today.
Method number 1 is gene cloning, which honestly acts like a biological cut and paste. We start with isolation, where we use restriction enzymes to essentially snip out the exact gene we want. Next up is ligation. This is where DNA ligase acts as the molecular glue to paste this gene into a circular piece of bacterial DNA called a plasmid, creating what we call recombinant DNA. Then comes transformation, where we literally insert this new DNA into a living cell, usually a bacteria. And finally, selection, where we grow the bacteria and pick out only the ones that successfully took in the new DNA.
Our next tool is PCR. Think of this one as your biological photocopier. It relies on extreme temperature shifts to multiply DNA super fast. It starts with denaturing. We heat the DNA up to snap it into single strands. Once it's cooled down during the annealing phase, we add short primers that bind right to the target sequence. Finally, in the extension phase, a heat-resistant enzyme called Taq polymerase comes in and builds the new strands. And then, this cycle just repeats, copying the DNA exponentially.
Okay, let's dive into the absolute revolution of modern biology,
CRISPR/Cas9. Believe it or not, this tool was actually discovered in the immune system of simple bacteria, which use it to fight off viruses. Here's how we hijack it. Scientists create a piece of guide RNA, or gRNA, that exactly matches a target DNA sequence. This guide RNA leads a protein called Cas9, which acts just like a pair of molecular scissors, straight to that specific spot on the genome to make a highly precise double-strand break. From there, we can turn off a gene or even insert a brand new healthy sequence. It is programmable, it's precise, and it's incredibly powerful.
Section 5. Mutation Misconception Check. Before we wrap up, we need a quick Mythbusters check to clear up some major misunderstandings.
Okay, the first major misconception is the belief that all mutations are harmful and inevitably cause diseases or abnormalities. That is simply a myth, guys. The fact is, mutations can be completely neutral. These are called silent mutations, and they don't change the organism's traits at all. And importantly, mutations can sometimes be highly beneficial, actually giving an organism a brand new advantage in its environment.
The next myth is that you need some crazy radioactive spider bite or a toxic chemical spill for a mutation to occur. While chemicals and radiation absolutely can cause mutations, the crucial point here is that mutations also happen naturally and spontaneously all the time. Just during the normal, everyday process of DNA replication inside your cells, tiny typos can occur without any external influence whatsoever.
The final myth we really need to bust is the idea that mutations are somehow separate from the process of evolution and that evolution is only about adaptation. This highlights exactly why understanding genetics is so absolutely crucial. Mutations are actually the essential raw material for evolution. Without spontaneous mutations creating genetic variation in a population, natural selection would literally have nothing to select from.
Section 6. Formative Check and Recap. Let's see how well we've absorbed all this information today.
First question for you to ponder. Think back to our stickleback fish. How does a targeted typo in a tissue-specific switch differ from a typo in the main coding region of a gene? Well, remember, if the coding region is broken, the protein is broken everywhere in the body. But if just a switch is broken, like that pelvic switch, the fish only loses its pelvic spines, while the jaw develops perfectly normally. It's all about location.
Second question. When we talk about biotechnology and CRISPR, what is the specific role of the Cas9 protein? Ask yourself, what do the scissors in our CRISPR editing kit actually do? That's right, once the guide RNA leads it to the exact right spot, the Cas9 protein is the enzyme responsible for making that highly precise double-strand break in the DNA.
To recap our whole journey today, we learned that genes are masterfully controlled by specific regulatory switches. We saw that humans can now literally edit this biological code using incredibly precise biotech tools like gene cloning, PCR, and CRISPR. And we confirmed that mutations aren't just harmful accidents, but rather the natural driving engine of genetic variation and evolution. You now hold the keys to understanding the biological instruction manual of life. So, what genetic mystery will you explore next?